In today's video, we'll be talking about dumb RNA at the beginning of life. Hi, my name's Eduardo Hernandez. I am the creator of Emergent Chemical Evolution, also the author of the book by the same name. You can find the book at all major online retailers. Welcome back, my natural-born scientists. Dumb RNA at the beginning of life is not that it's a dumb idea, because they were definitely there. What I refer to as dumb RNA is RNA segments that had no information, no coding for proteins, no information that for self-replication. These were just random strips of RNA that basically just fumbled around in the primordial soup. The action of the chaos engine would cause those RNAs to continue to be swept away with the currents and taken through pH gradients, and they would do a number of things, one of which would be cleave themselves or each other, because that's one of the main catalytic abilities of RNA, is to cut. Right? That's all they do. That was a major catalytic property of RNA that was discovered back in the 80s. And this realization led them into some broad assumption that RNA would be able to catalyze its own self-replication. That I find, again, like I've said in my other videos, untenable. What I do find possible and is evident in the structure of the RNA segments used by life today. One of the most important RNA segments used by all life is transfer RNA. Transfer RNA is a short strip of RNA that is replicated from DNA and is the template for any proteins created by the ribosome. Now, like I mentioned in my previous video, the ribosome is just a multiplication of viroids. Viroids is one of the main products, I believe, of the chaos engine, which was the beginning of life. I named the viroid as the first life form. That to me makes abundant sense, since it does not encode for any protein. All it does is hijack the components of RNA replication and make copies of itself. That's it. It doesn't code for a protein coat. It doesn't code for a protein to replicate itself. It just replicates copies of itself. Of course, RNA being a very error-prone molecule, when it's replicated in such manner, with almost every replication, can't say that unilaterally, that all replications will introduce errors. But one of the most common errors in the replication of any nucleotide is the introduction of multiple copies of itself, or multiple copies of singular bases, making that RNA chain a little longer, or eventually doubling in its size. That's one of the things that can happen. Is also something that would be uncontrolled at the beginning of life. There will be no proteins to error check the copies of these strips of RNA. There will be no systems of correction, no systems of rejection. So these RNA segments would multiply unhindered because there's no competing molecule for these resources. It is the sole molecule that would control the nucleotide market, let's say. These would not be in competition with any DNA organisms. DNA would not have been invented yet. They would have full reign of the environment, of the aqueous environment that is the ocean back then. And they would have been trillions upon trillions upon trillions of these segments made. And that's why I believe we still find them today as viroids. They, like E. coli, like other organisms from primordial times that continue to exist today because they were able to find themselves in a niche of the environment that either changed very little or hadn't changed at all in all these millennia. So to me, it's no surprise, especially after so many copies of viroids being made, that we continue to find viroids today. Now, the important thing here is to see that viroids are a strip of nucleotides that ranges between 375 and 390 nucleobases. This number of nucleobases is key, I believe, in forming a stable nucleotide that's unprotected by a protein coat that could be robust enough to survive the jostling and violent environment that would have been the Hadean era oceans. That being said, they would be, of course, the starting point of transfer RNA. When they would autocleave, they would take the large 300 and plus 375, 390 nucleotide nuclear RNA and cut segments of itself off and make it small enough to then take the conformation of a transfer RNA. At the same time, it would also, during its unhindered replication, it would also have the era of making multiple copies of itself because there's no check, no system for error checking or restricting the size of these RNAs. They would henceforth create very large segments of nucleotides and would eventually be large enough to take the conformation of a pre-ribosome type RNA molecule, where it's large enough to take RNA, take transfer RNA into itself, and then use the catalytic nature of RNA to cut off any of the any of the amino acids that would have been attached to it. This system would have readily come up. You would have had strips of RNA attaching itself to amino acids. I mean, amino acids would have been ubiquitous in the environment at that time, especially during the heavy bombardment, because all these meteorites would be laden with amino acids. So being that the chaos engine was in place and working, taking amino acids, making them longer, making them react with each other and on other amino acids to therefore eventually create RNA segments that would be long enough to take the conformation of viroids and allow it to take a conformation that was robust enough for those segments to survive, and then going through the pH gradients, find itself in a gradient where the right amino acid polypeptides would have united, take a conformation that would allow the replication of RNA. And of course, this would be a very simple replication mechanism with no error checking, no feedback in order to stop working when it's made enough RNAs. There will be none of those checks. These polypeptides would just start doing what they always do, which is reacting with amino acids and creating novel proteins, which will be the source of another video, have the the conformation of those amino acid segments would readily lend themselves to form these kind of proteins that would easily replicate RNA. We have at least 20 different versions of these type of polypeptides in nature today in the different kingdoms of life. And they, again, take multiple forms. You have ones that take many small polypeptides and unite to do the function of replicating RNA. And then you have in higher life forms that evolved larger proteins, single segment proteins. So if we were going backwards, and again, what we see with simple life is small segments of RNA taking conformation and combining and doing these actions, it's only reasonable to expect that to have occurred during the primordial stage with the chaos engine in place and working. RNA would have initially would not have had any information. It would be dumb segments of RNA, unable to control any other biological reactions or have the information to self-replicate. Self-replication of an RNA segment there is no representation of that in modern life. There is no evidence for RNA to have ever self-replicated. What there is evidence of RNA working in conjunction with other proteins, other RNAs, always working in concert with other molecules to do its job. And I feel, excuse me, that the chaos engine and the emergent chemical evolution created the perfect environment for those molecules to have converged and coalesced into a system that initiated life. So dumb RNA is what should be expected at the initiation of any origin of life theory. The reverse engineering of proteins, as in the other style of metabolism first or membrane first type of origin of life theories, are to me untenable. So dumb RNA is the expected product of any origin of life theory, even if mine doesn't turn out to be correct. That has to be the initiating molecule, a dumb RNA. There's no way that an RNA would have been complex enough or large enough to be able to replicate itself. At least ad novum. But yeah, dumb RNA are our ancestors. So I hope you enjoyed this video. I know I'm a little down. It's really early in the morning, but I promise with more videos that I make, I'll be more energetic, spontaneous, at least more entertaining. So please hang in there with me. Please subscribe, like, tell your friends about my theory, about my videos, about my book. And hopefully I'll be able to convince you by the end of these series of videos that my theory is correct. Thanks.